Hello, my name is Ravi Vij. I'm from My Cancer Haven, and today in our conversation with Cancer Expert series, I have with me Dr. Hans Lee, a rising star, associate professor, and director of the Multiple Myeloma Clinical Research Program at one of the top cancer centers in the world, MD Anderson. And we're going to be talking about Celmods, a new class of drugs that we expect to be on the market in the future for myeloma. So welcome, Hans.
Thanks so much. Thanks, Ravi, for having me on the program.
So Hans, I guess to start off, can you tell our audience about what cell mods are and how do they mechanistically seem to work?
Yeah, that's a great question. So Celmods are basically molecular glues, and they're in the same class of drugs as previous iterations of myeloma drugs that we're familiar with, lenalidomide, pomalidomide, and thalidomide. And essentially what they do is that they bring together this complex on the left part of the slide, this E3 ubiquitin ligase, and basically bind to this molecule called cereblon. And basically in doing so, it recruits proteins that might be important in the growth of cancer cells, in this case, Ikaros and Aiolos in multiple myeloma, and basically allows these proteins to be tagged by this other protein called ubiquitin, and basically leads to degradation of these key proteins in myeloma growth, and hopefully leads to myeloma cell killing as a result. And one of the interesting things about Celmods that potentially differentiate them from their previous iterations, lenalidomide, pomalidomide, and thalidomide, is that they're more potent. And so essentially, they bind to cereblon more tightly. They can also induce greater potency in substrate degradation of Ikaros and Aiolos. And so what we're seeing is that they have activity in patients and in laboratory models that are resistant to lenalidomide and pomalidomide, for instance.
So obviously, Celmods are cereblon modifying drugs. You're saying that that's where the target. So I guess in clinical development, we've seen two that have had data presented, and we'll go through this in the next few slides. So can you tell us about iberdomide? What do we know about this drug? And then we can talk about the other one.
Yeah, so iberdomide was